Janet's fourth and last talk. You have the floor, Janet.
All right, 50 minutes, yeah. So Beatrice had asked me at lunchtime to actually show you a picture of Quattrocienigus, and this is a picture. This is in the middle of the Coahuilan Desert. It's associated with a little town that has about a population of about 10,000. This, of course, is one of the mountain ranges. The town is off to this side. This is one of the common desert plants there called sotol, and they used to make spirits out of. And this is an intermediary lagoon. The spring is down this way, flows through here, and then out to the evaporative pond where the gypsum dunes are. All right. Hope you all had a good lunch. So we're going to have one more. I'm sorry, I have to show you one more tree, but this one I'm going to go over fairly quickly. And I want you to be thinking about how to apply this to the origin of life, but also what might be wrong with this tree. Okay, that's really what I want you to think about. Come on in. All right. So when I was a young grad student and I was looking, I've already told you that I was terrible in the lab. I mean, I was a disaster. So of course they're trying to figure out somewhere to put me, so they put me in front of a computer. You can do less damage there. And so I'm looking, not only am I terrible in the lab, but I have difficulty sequencing, all the technical abilities that go along with that. And frankly, I think there's magic in molecular biology labs. I think you hold your mouth a certain way and then things work out. And I never figured out how to do that. So I just started doing gene trees and started doing phylogenies, basically gene histories. And so I was working mainly with bacteria, and something began to show up in bacteria. Now, I'm not talking about on the 3 branches of the tree, not talking about archaea. If you believe eukaryote comes out of archaea, I'm not talking about them. I'm just talking about bacteria. So I'm taking 16S ribosomal RNA, and I'm looking at those trees. Now, how many of you, comparing the phylogeny, comparing the history, the family history of them, how many of you have ever seen a bacteria under a microscope? 2. Okay, I'm going to tell you, they are the most uninteresting things you've ever seen. You look under there and you think, I don't know, is it a bubble of water? Is it something alive? It's not like, like amoebas. If you can go, probably could go anywhere in any street, take a sample, put it under a microscope, and you can see an amoeba. Amoeba is like what the 1950s and 60s horror shows were about. It engulfed, it kind of moved around and engulfed. There's no bacteria that look like that. I mean, they're either one thing or the other. They're either spheres or they're rods. Sometimes they actually divide in a strange way that you can get a little bit more identification. There are a few that kind of have a spiral look to them like this. But for the most part, the staph in the back of your throat is just a little coccus, round shape, cocci. Bacillus is always rod-shaped. So basically 2 shapes. Well, what started to happen, so we've already talked about how misleading morphology can be. Remember, we talked about the 5 or 7 domains of life, and that was all just based on morphology. And it really misled us to characterize things properly. So once we had the DNA, we could actually characterize them properly. Well, bacterial morphology, if anything, if 90%, I'm just taking a guess, 90%, if 90% are either cocci or rods, how are you going to separate them? And you would think that if you wanted to think about the last common ancestor to bacteria, it has to be one or the other, right? If the last community, if the oldest community of bacteria gave rise to all these others, and you have this mixtures of morphology, that one back there had to be some shape. What shape would you guess it would be? A triangle, not an option. A lot of the origin of life experiments look for They look for a circle. I mean, it's you know, you're going to have something you can make into a blob, a circle. It's what? That's there's nothing simpler. I mean, you can make. Okay, well, he actually brings up a good point here. So we're constrained by something. So we're you don't need to see this again, or this one, but you do need to see this one. So all life has membranes. All of life has membranes. They're phospholipids. All of bacteria have cell walls. So they're not blobs. They're constrained by a cell wall. And so you know the last common ancestor of all of bacteria either had to have probably a rod or a spherical shape, and it had a cell wall. So that's how uninteresting they are. You know what this one is, right? This one's the one that's a little bit different, but what is it really at its basis? It's a rod. But this is what the cocci look like. All right. So that cell wall is made, excuse me, that cell wall is made of peptidoglycan. It opposes internal hydrostatic pressure and it prevents that membrane from rupture and death. It has a role. Sorry. Let me get a drink real quick.
That's what soap does to bacteria, right?
Right. All right. All right. So the perspective was then, what I started doing, you already know this, what I started doing was I thought, well, is there any kind of pattern if we look at all of bacteria, we look at their family tree, is there any kind of pattern, can we put on top of that tree what the morphology is? And can we get at the fact that probably it's going to be a cocci? So here's what it actually showed. The most deepest branches were all rods. The oldest morphology looked like it was rods. Totally unanticipated. Here's another look at a tree like that, just so that you can get the idea. So the farther out on these branches you go, the younger the organism is, it's internal to this one. And blue means that they're rod-shaped. And so you can see that everything looks like it started out as a rod shape. These are some of the oldest bacillus that we know, some of the oldest in the family bacillus that we know. All right, so the summary of the results were that rods were in the deepest branches. The cockle phenotype evolved many times and persists. Some morphologies, like spirochetes, and there's the blob ones, mycoplasms, are highly unique. Filamentous forms of bacteria are found throughout the tree. That's a separate way of cell division. And helical forms are widespread in the gram-bacteria. Don't need to know those particulars. You just need to realize that once we map the morphology on top of the 16S tree, contrary to what we thought was going to happen, it looked like rods were the most ancient morphology. All right, so what you had to then, so if you're a scientist and you've discovered this and you know that there's going to be some pushback because it seems like the simplest thing should be a cocci, what are you going to do? We have all these gene sequences happening. We're starting to understand how cell division happens. So why don't we look at the genes that actually cause cell division and see what happens in the cocci forms and in the rod shaped, and see if it fits, if the genetic information outside of 16S fits with this idea. So that's what we did. So it turns out that in order to be a rod, there's a balance between elongating your cell and then getting to a point that you need to divide. It has to do whether or not that happens. So let me explain this. If you're going to retain the cocci shape, you can't get longer. You have to stop getting longer way before you get out of the circle shape. All right. So it turns out that the rod shape is important in antibiotic resistance, and it aids filamentation. So a different way that bacteria can divide and still remained a long filament. So that helps in ecosystems. So there was a selective advantage to being a rod. All right, so our conclusions were a coccus appears to be degenerate rods. Once you get the fixation of rod morphology happened early, could have been an adaptive process or just chance, could have been merely a result of the physics of peptidoglycan as the constituent for the cell wall, but it most definitely was the winning strategy for combating osmotic pressure for bacteria as we know them today. It's very consistent. Coccus was likely, that's actually wrong. Coccus was not the first. That's not, somehow I messed up. It should have had not likely the original shape for the bacterial lineage. So what does peptidoglycan do? So one of the things then you got to think about, if peptidoglycan is the thing that's controlling how the morphology evolved all through the bacterial kingdom, what does peptidoglycan do for that first cell? What did it do? And it had to be in the first cell of bacteria because it's in all of them, majority-wise. Well, it follows a maximum membrane to cytoplasm ratio, so there's no need for internal membranes. If you look at a eukaryotic cell, there's all kinds of internal membranes which allowed complexity to occur as cells repeated and became more complex. That's not the case in bacteria. Allows it to reproduce rapidly, provides a scheme for adaptation, evolutionary diversity, controls the interaction with its environment, and it allows bacteria to stay small. So once you can have internal membranes, the cell can get bigger and you can transport things around through those internal membranes. Bacteria don't have that option. So because they stayed small, they have to stay small because of this peptidoglycan layer. All right, so there was the cellular evolution of the three main domains of life had different solutions to solve the problems of getting smaller, getting bigger or staying smaller. So in bacteria, it was peptidoglycan. In archaea, it was pseudomurine, a different kind of cell wall or cell wall type structure. And in eukaryotes, it was cytoskeletons and mechanoproteins. Charlie's going to give a little bit more information about this tree being really just two branches. And one of the things that I would push back on him if I'm listening to his talk is the fact that there's actually three very different ways that cells use to solve the problem of maintaining their morphology and the internal structure. All right, so in the last decade, the genetic underpinnings of cell shape and division have really come to fruit. So we know that, again, these are just the acronyms for the genes that do certain things in an organism. So the tubulin in a eukaryotic, these are eukaryotic proteins. The tubulin is part of the structure that allows constriction and muscles. In prokaryotics, the prokaryotic similar protein is FTSZ. In eukaryotes, there's actin for muscles. In the prokaryotic world, they use a protein that's just got the acronym MREB. So this is just a schematic of how these two proteins actually work together to create a structure that allows a division. And the only reason this is important, the only reason you have to even think about this is what underpins that finding that we found, that rod shapes were the first. Because once you have an organism that does this, if it resorts to being a coccus, it can no longer go back to this shape. It's in a dead end. That's all you have to know from this graph. Same thing here. I don't think we need to do this one. We won't worry about this. There's no true cocci-shaped cells that have any MREB homologs. So all of that internal structure that lets you maintain that rod shape has been lost in every cocci lineage. It can never go back to the rod shape. So it's a dead end. So the genetics, the point of this is the genetics actually supported what we had found. All right. The applications to the origin of life are for bacteria, the 60-nest ribosomal RNA backbone provided a historical scaffold for the bacterial lineage that allowed an accurate evaluation of the evolution of cell shape. And cell shape sounds trivial, but it's important when you're trying to figure out how to contain whatever you need to contain in an organism. I don't think we'll worry about this or this. So what is the selective advantage of the shape? What's the selective, why did the first bacterium, why was it a rod shape? Well, there's a physical advantage because of the volume to surface ratio. There's a polarity assignment, which is good when you're trying to separate into two cells and you need the equal amount of everything in both cells, so that was important. A little bit difficult to do that if you're a caucus shape, right? You don't know where the poles are. And then there is some indication there was a predator and prey advantage. We're not going to go over these. So here's what I'm going to do. I was trying to think about this. Y'all have been really good to listen to things that I know that are so far outside your field and really don't have a lot of application maybe to astronomy. But this is a great example about why in the origin of life we have such a difficult problem of taking extant life and going backwards. So here's my example. Anybody know what this is? This is my analogy. We can only push it so far. Anybody know what this is? I see some shaking heads. You have one of these in your home? Anybody have one in your home? Nope, nope. I don't see anybody saying yes. I actually have one at my farmhouse. Doesn't work. It's just it's just for you to look at. You know what it is? What is it? It's a telephone, right? I don't know how old this is. I mean, 100 years at least. It has no dial. How in the world did you connect to anybody? Anybody know how that happened? There was an operator, yeah, and what'd you do? Well, actually, I didn't do this. I'm not that old. But you had to actually click that little, where the thing that you listened to, you had to click that twice, and that would alert the operator that you were trying to make a phone call. There's no way to dial anybody, right? Okay. This is one of the earliest telephones. Looks a little bit, you could find some similarities between this one and some of the others. Huh? I think this one's earlier here. Here's that same one. Here's an improvement, right? Yeah, it's got a dial on it. Here's one that's really fancy. I don't know, it's got 3 buttons at the top. I don't know what they do. I am old enough to remember that when you would call somebody, you had to listen because we had something called party lines. And that meant that somebody else might already be talking on whatever the line you were on. So you had to say, excuse me, I have an emergency. You have to get off the phone. It's my turn to talk. That's hard to believe, isn't it? Here's another one, a little bit later, right? You used one of those? There might be, who has one of those around in their house? Not around? Okay. 50 years ago. What are we looking at here? What are we looking at? Yeah, we're looking at telephones, but what are we looking at in sort of, if we want to kind of bastardize the word, we're looking at the evolution of telephones, right? I mean, you can see some common things here. All right, what about this? Can you see that? That's your grandmother. She was actually the first job I ever had after I got married was my husband and I, he lived in Chicago. I moved up there with him. And you might be surprised at this. He thought I was a little bit kind of a nervous person, you know, and needed something to do. So he got me a job as the switchboard operator at the Missouri Pacific Railroad lines. So I actually didn't look quite that old, but, you know, I pushed those things in and connected people. Sometimes I wasn't very good at the job either. What is that? Photograph. So what I'm trying to lead you to here is we're seeing an evolution of phones, of connecting with phones. And at the same time as some of this was going on at this time, another thing was happening. Another sort of technical evolution was happening. So on the railroad, we used to have a way that we would, my dad was a switchman on the railroad, which meant you actually told the cars to go down tracks or unhook them so they could get on another train, you know. So they had these things called hump yards, where you'd push this load of cars over this hill, and once it got to the top of the hill, you would determine, there were like 25 rails out that, and you would determine which rail they had to go down in order to make a new train to go to a new position. And it was a big deal when they got the first computers in to actually handle that. You know how big they were? They were in this room that was like half the size of this, and you had to keep it down to 60 degrees because they were so hot. And all they were doing was just switching which way the train went. So that's a computer. What's that? Anybody got one of those in their home? Do you try to sell it on eBay? So at the same time that phones and communication were changing, we had a whole nother industry that was changing, that was computers. And that developed extremely rapidly. And then we had cell phones. Anybody ever use one of these? I never did. Probably nobody had enough money to do that. How about this one? No? Yeah, I see a yes. This one? I personally love this little Nokia. I think I still have this one back at the house. I mean, it's great. I mean, but these are beginning to look a little bit more like each other, right? But the trick is, huh?
They're rod-shaped.
They're rod-shaped. Yes, they're not caucus-shaped. They lost that. Okay, so now how many of you have one of these? Raise your hand if you have one of these. Okay. Let's pretend that Wikipedia rewrites history and gets rid of all those things I showed you at the beginning. And all we have to look at to do phylogenetics on, are these right here. So you can take this apart. You can try to get it down to its most primitive parts. You can try to figure out how that horizontal gene transfer event came in that was computers, that had nothing to do with the beginning ones that we ever had, that one that you have to click, that didn't have a computer in it anywhere. Do you think you would ever arrive at that very first telephone that I showed you? It's impossible. Why? The trajectory was lost. I have a suspicion, it's not something I like, and I'm not a negative Nelly, actually, although you might think I am. I think that whole thing I showed you about morphology, I think it's accurate, I think it's good, but I have no idea where it came from or what was before it, and I don't think I have any shot at finding that. Once we had this kind of thing in telephones, unless the world ends and something happens and we have sort of a resetting event, we're never going back to those other telephones. And I don't even know this might not even be relevant in another 100 years. Maybe you have some chip inside you that, you could just walk up to somebody and you don't have to talk to them. I don't know. My point is, in science, we try to do the best we can, and when we're talking about origin of life and when we're talking about things that have to do with how evolve happens, I think a lot of what we're looking at 4.2 billion years later are processes that have become so valuable and so integral to where we are that getting back in time is really, really hard. I mean, if you actually think you could get back to what that other phone looked like by looking at these, you need to come over to my field. Does that make sense to y'all? Okay. Okay, so just for fun, because this is the last session, and I didn't want to give you any more trees or any more protein acronyms, I thought we'd talk about artificial intelligence. And the only reason I bring this up in an origin of life talk is because we've been trying to talk about past, we've been trying to talk about present, so now we're going to try to talk about future. Any of you ever talk to Siri? She's your, artificial intelligence pal. She listened to you well. She answers, you can ask her some interesting questions, right? I mean, I saw a talk one day that somebody said, Siri, are you alive? And she said, maybe go to the Genius Bar to answer that. Artificial intelligence, though, there are people that think that's somehow going to supplant and become another form of life. Now, I'm going to be honest with you. It's difficult for me to imagine that. But it's actually fun to, in a group like this, to think it through together. So I looked up, I thought, well, are there any scholarly papers on the evolution of artificial intelligence and how it might actually take off? And lo and behold, there was one. But before that, let me entertain you with...
We designed them to be trusted with our homes, with our way of life, with our world. But did we design them to be trusted?
The rollout of USR's new generation of robot, the NS5, was marred by the death of designer Alfred Lanning.
Identify. Murder's a new trick for a robot. Respond.
He don't murder him!
We're gonna miss the good old days.
What good old days?
When people were killed by other people.
My robots don't kill people. That thing threw somebody out.
All right, enough of that. You get the idea, right? These are thought experiments. You guys are young people. Y'all have got, you can think outside the box better than somebody like me who's got all these, ingrained prejudices about how things work. What do you think about artificial intelligence? Is it ever going to be able, what was assumed in this particular movie? Did anybody see the movie? Yeah, what was assumed in the movie? Can you answer? I'll take it up there.
Decided that we needed to protect us and took over what we're doing so we cannot hurt ourselves. That's it.
Yeah, so it actually was able to determine good and bad, right? There was this whole morality part to it. But if you don't want to think about the morality part, it could make decisions on its own, right? It developed a consciousness that could make decisions on its own. Now, that's sort of something we talk about when we talk about humans. But let's talk about, when we try to define life, if we're trying to define life in AI, what would that include? Well, I did find an article, The Past, Present, and Future of Artificial Life. Let's see, it was in 2014. And they set out about 10 parameters that they felt like artificial life would have to contain in order to be considered life or alive. You understand the connection I'm trying to make. We've been studying the origin of life, so now we're going to talk about a whole nother different kind of weird life that we actually make ourselves. So one of the things they talked about, and we talked about this at lunch, was autonomy. So an artificial intelligent being has to have a bounded network of processes that self-maintains. Seems reasonable, kind of a good definition of what we can do. It has to be able to self-organize. Phenomena where local interactions lead to global patterns or behaviors. Pretty sure we've got AI that can do that now. It has to be able to adapt. It has a change in an agent or system as a response to a state of its environment that will help the agent or system fulfill its goals. Now that's a little bit more geared towards thinking of artificial intelligence as sort of human replacements. But still, the idea that it has to be able to adapt, we clearly know that origin of life on this planet had to learn how to adapt, right? So if you're going to have a separate entity that becomes some kind of organism that's alive, it's going to have to adapt. Has to evolve. So computer science has exploited artificial evolution extensively, initially with genetic algorithms. That's a done deal. And it has to learn. Artificial neural networks are a well-known approach to learning. So how close are we getting to making AI, and we are making it, right? We are the ones, but it becoming separate from us and being able to become a live organism. Now, let's just, I mean, we're really thinking outside the box here, trying to make this so that, you know, it's interesting this last part of the day. What value would it be to have an organism, an AI organism, if you're doing space travel? What? Ostensibly, it's not going to have a physical body that decays. That means, you know, time travel doesn't create such the problem it does before, right? You might have to have some kind of, you don't have the same kind of worries about radiation exposure.
It could wake up the cryogenetically preserved humans on the space station.
Yes, it could. It could be, unless it decided it didn't want to. You know, we do have that. You know, maybe they've figured we were what does this have to do? You know, there's been I'm with a team out of England that kind of surprises me. England has this, there seems to be this sort of interest in extraterrestrials and UFOs and stuff. I mean, we're even doing that a little bit in the U.S., which is, has anybody been abducted here? No? Okay. I'm surprised how many people, actually, if I'm not going to ask because I don't want you to have to reveal anything, but I'm surprised at how many people think that UFOs have been to the planet here. I'm surprised at how many people, you know, so I always like to see what the odds are if people think there are there is intelligent life on other planets, you know, and the people that don't. If you can have a AI that can actually travel, and we can envision that, then why couldn't a more advanced civilization somewhere else have an AI? Why haven't we had anything come towards us like that? That's a whole different type of life form that was actually an artificial intelligence and went to breach that place where it actually became live on its own. They're just kind of thought experiments. But I brought this up solely for Charlie and for another person we've been talking to, because we've been talking about, well, what is a mind? So you might wonder, I guess the point that I'm trying to make here is if you have an artificially intelligent being or organism, it has to be able to think and consider things. And so there was an argument at lunch. Do you want to talk about that a little bit, about what you and...
Yeah, we didn't have a discussion, we just had an argument.
Yeah, I know. We'll tell the argument.
Oh, well, actually, I was talking to this. Well, basically, my wife and I are writing a book. We don't believe in free will, and so a lot of people think they have free will, and a lot of materialistic reductionist people think, no, we don't have any free will. So I mentioned that to Gustavo's wife, Andrea, and she's a psychologist, so she thinks there is free will. And she said, yes, there's environment, and there's development. No, there's DNA. I was saying there's DNA and an environment and development, and that's it. There's nothing else. And she said, oh, yes, there is something else. You have DNA, development, and that produces a mind, and the mind has free will. And she said that, and I said, oh, I disagree with that. And then I told her about self-driving cars. I said, do you think self-driving cars have free will? Because they can make decisions about where to go. And she said she would think about that. And
okay, so the point that I'm bringing this up is AI going to have free will? Are we going to have this? Would a community of AI, in some way, if they're primitive, of course, they're not going to, if they're, you know, just cellular or something, but if they're actually doing work, if they're following any of the guidelines of this kind of, these kind of characteristics that AI would have, it's a wonder. Do you come up with what Andrea thinks, that there's more to that, to us, than just our brain and we have no free will? Or would an AI actually just be, you know, an automaton that in this situation, like Hal, I'm going to do this or this or this? Or would there be an element of free will in that? They're just thought experiments. They're not meant to mean anything other than to just get you to think and maybe have you participate. I already mentioned it's an advanced way of life that is not hampered by physical decay. So what do you think? What do you think about AI? Have you thought about it?
Janet, I think we must make the distinction between intelligence and conscience. Intelligence is trivial. The cell phone is intelligent. It solves problems according to a predetermined set of rules. Conscience sometimes creates rules.
And I was trying not to make this I was trying not to make the leap into, what we're bordering here is on what science can do and then what philosophy helps us understand. And I was trying not to broach that. I was kind of trying to hedge around it, dance around it a little bit, because I think that's a totally different argument that is probably more difficult in a science conference. But it is exactly where we finally get to. I mean, how do you teach? Let's assume that we can have an AI that has all of the things that make it seem like it's alive. Is it going to develop a conscience on that, just on that alone? I mean, I don't know. I mean, certainly we have consciences, right? Certainly we know when we see something terrible on Facebook. I mean, everybody in this room is just gonna, put their head. I mean, the killings in this week in the U.S., I mean, there's not a single person in here that doesn't think that's awful. But are you ever going to be able to have an AI machine that you just say, okay, anytime this happens, then you need to put your head down because it's awful? So the whole idea, I was trying to step away from that, but still try to bring life in. Because I don't think bacteria have a moral code. Right?
I disagree with almost everything you're saying.
Well, of course you do. What do you disagree with?
So there's something called Darwinian psychology.
Yeah.
And the idea there is that when we have moral opinions about things, and for example, say there were two tribes. There's one tribe thought that, I don't know, that they were important, and another tribe said, well, I don't know if I'm important or not. And well, this tribe over here thinks they're important, and they will start to do things to preserve themselves. So, or imagine there's a mother. One mother thinks, oh, my darling baby's the best. She does a lot to protect it. The other one says, oh, I don't know, it's a baby, I don't care. And who will survive? The survivor will be here. So the whole point is that the moral feelings we have, the moral responsibility, is something that has evolved. This is called evolutionary psychology. So the point is that this morality that you want to say depends on a mind is something that is easily selected for and can be explained without having to think about it.
Well, anybody have a pushback on that?
Yeah.
Anybody?
Well, the point of that story is, though, if you can evolve it in this piece of meat this big, you can evolve it in a piece of meat of any arbitrary size, like chimpanzees or even flies have. I would suggest that almost all life forms have some type of morality that has been selected to constrain their behavior in ways that is conducive to survival. And that's what I would stop.
So are there any exceptions to that you can think of?
I think there is a turning point when you have this biological system that's capable of abstractions. That is to act according to things that do not exist. They only exist within the system.
Right.
They have no objective reality existence whatsoever. That's a turning point. Charles doesn't agree, of course.
Let me tell you, there are. So I'm gonna stand firm on the fact that I don't think bacteria have a moral code. Sorry, I'm gonna go there. But I do. There are slime moles. I don't know if you know what a slime mole is. Just imagine. I think the words are fairly descriptive of it. And when it gets under stress, then there are a certain group of the cells that will sacrifice themselves to make stalks to go up. So there's that sacrificing for the good of the community. I don't know if any of them are crying inside because they had to die, or sorry that.
Eukaryotes have morality, but prokaryotes don't.
Well, I don't see any evidence they do.
Well, you just gave an argument for
survival. That doesn't have anything to do with whether or not something's good or bad.
Well, sure it does. That's have everything to do with it. That's the only thing that has anything to do with that.
Okay. Does anybody else want to jump into this argument? See, I need to not be quite so nice when I answer him. See, he's so, he's taller than me and bigger than me, so, he's using unfair advantage there. Anyway, it gives you something to think about, right? It gives you something to, who knows where we'll be in 20 years? I mean, I'm not trying to in any way convince you that we're going to have artificially intelligent silicone machines. Your phone's not going to be, jump in there and decide it's a living organism. I'm not saying that. Oh gosh, you're gonna disagree with that?
No. I'm just saying that a similar thought experiment would be when people start to present examples of life forms that they've created in the laboratory, not just minds or anything sophisticated like artificial intelligence, but a cell. For example, Craig Venter says, he takes the mycoplasm and he one by one does a knockout experiment on the genes and says, okay, I'm going to make the minimum amount, and then I use chemicals to make the thing, and then he says it's kind of artificial life form, but it's kind of not.
Right.
So then you could ask the question, is that a life form? But I'm not sure that's that important.
Well, part of the reason why I included this is, if we've been talking about life way back, however long ago it was that it started on this planet, and we are trying to figure out what it looked like, and maybe it looks like everything now, and maybe it doesn't, nothing has stopped. I mean, we're continuing to evolve. We're continuing to change. And just for right now, it looks like one of the things that people are thinking about is artificial intelligence. I mean, it is getting really, really smart at doing a lot of things that we can do. So all I'm trying to do is just get you to open your mind a little bit in a different category. That's all. And you can, I hope you're going to come down on my side and not his. Bacteria don't have a moral code. I do think they scream when we try to put exogenous DNA into them. I do think that's happening. All right, so I hope Google Translate, my artificial intelligence vehicle, tell the right thing. What I'm trying to tell you is thank you for letting me be here. And I hope that somebody or all of you in this room make a substantial contribution to science in your field. You have that obligation as smart people to do that. And you need to open your mind, talk to a lot of people outside your field so you don't get stuck in one place. But do the best you can and work hard and follow the money. That's it. Oh, if you want questions, I don't know if you have questions. You want to ask questions?
Yeah, we have a lot of time for questions. So we are joining questions from Janet's previous lecture into this one. So feel free to ask anything.
And nothing that you ask is going to be anything that, you shouldn't ask. Any question you have is a good question. And even if you think it's not a good question, it's a good question.
Hi, thank you for the talk. In the first horizontal gene transfer, is there a guarantee that the second organism will get the information released? I don't know if you remember the slide, but
this was from the morning talk.
Yes.
Okay, tell me again.
There was a first organism releasing information to the second organism in the first horizontal gene transfer. Is there a guarantee that the information released in the environment will get to the second organism, or is just blindly releasing information? Yes,
that's a great question. From my understanding, I think it's a stochastic process. I think it just is a matter of happen chance. And I think, and this may be a prejudice on my part that somebody can actually question, I think it takes lots and lots of opportunity for something like that to happen. Now, I'm not saying it takes lots and lots of time. I have no idea about the time. I really don't understand. You had a very complex, it looks like from the geologic record, you had a very complex photosystem apparatus in place really early. I mean, we're talking, that molecular machine is beautiful. And it's early, it's early. So it's just a matter of how fast things happen and how many options you get. It's winning the lottery.
Thank you.
Somebody wins the lottery every time we have a lottery. So, it's not improbable. Yeah.
Indeed. Thank you.
More questions?
Regarding the morphology of the cells,
Yeah.
you mentioned that cocci, if they're round, but when they divide, they have to elongate to divide, right? They have to become.
They don't really elongate. So the way that has to do with the way the peptidoglycan lays down. And they actually, that FTZ ring, which I didn't go over,
Yeah.
that actually gets in place,
Yeah.
and they don't really elongate that much. They grow bigger in volume. My recollection is they grow bigger in volume, but they don't elongate. They just get bigger, and
But
then
there's
it
that
pinches off.
FTZ ring, and that breaks this
Right.
spherical symmetry that you said would prevent them from enjoying, I guess.
Yeah,
I
but
guess that shape.
okay. Do they become like little mini rods at first?
Well, that's what they'd have to be if there's two spheres. You can't just get a big sphere and then it suddenly turns into two spheres. It has to elongate.
That's kind of what like Shostak's. That's exactly what happens mechanically. They don't become rods. You know, those experiments he did for containing them, they don't become rods
This
really.
big sphere.
They just squeeze. Yeah.
Well, squeeze in.
I think. I don't think they become rods. Okay, I know how to answer this. They don't become rods in the same way that a rod is a rod. They become a rod momentarily, very little bit, because that's just the dynamics of cutting a sphere in half. But it's not for the same reason. It doesn't become a rod. Yeah. Here's one. Good.
Então, segunda-feira teve uma palestra da doutora Evelyn. Ela disse sobre uma frase muito importante, que é evolução não volta para trás. Aí, esse negócio da perda de genes me fez repensar isso, porque o genes ele mostra o que é o passado evolutivo. E me faz... A minha dúvida principal é como que a perda de genes afeta a filogenética, porque a filogenética é baseada em se comparar com o passado. Como é que se compara com as perdas, sabe? E o que isso afeta naquele diagrama de mapa da vida?
He's referring to Evelyn's talk when she mentioned that evolution never runs backwards. But then you were mentioning that sometimes genes are lost.
Yes, that was actually...
He's asking about what's the importance, how does this possibility of genetic loss affect the whole phylogeny you're trying to build? What does it mean, information?
Okay, that's an excellent question. You understood the question. What happens? Okay, well, it's, from what I understand, and we've done a little bit of, there's been a little bit of work on this, but if something's lost, it's hard to find it, right? So it's hard to know. It's hard to know what you lost if you don't know what you lost. But what happens is we have remnants of genes that they become something called pseudogenes. So when you do you do the genome of an organism, and you're looking at all of the coding regions, and I'm talking about prokaryotes. I'm not talking about eukaryotes at this time. When you look at them, you'll see areas where there's a similarity for that area with a gene, but it has a whole lot of errors. I mean, it looks like it's decaying on its own. And what's happening, the reasoning is that it's no longer functional.
Are they expressible still?
They're no longer functional, so they're not being expressed. So the constant adaptive pressure that says you've got to keep this gene looking just like it was because it's not going to work otherwise is now off. And so the gene deteriorates, and you can see it as it deteriorates. It gets less and less like what it should look like, what it's the other gene it should be. And what that means then is that gene wasn't important to that cell. It didn't have it lost its reason for being, I guess. That's a little bit. It didn't have a function in the cell any longer. So you can track those pseudogenes. But once they get to a point where you can't tell that they have any similarity to another gene, then you don't know what they are. They're just pieces in the genome that, you have to just, I don't know what that is. sometimes you can track one. You know, I told you that if you had the GC content of a gene, you could tell it was horizontally gene transferred. You can also tell that if a gene looks like it's decaying because it's accumulating too many other, it's outside the percentage of the GC content that it should have. You can also use it for that reason too. But there's a selective, I'm not sure how strong this argument is, there is a selective advantage for microbes to have a genome that is manageable when you want to copy it and make progeny. And that is balanced with the fact, and that was what one of the slides I showed you earlier this morning, that is balanced with having enough repertoire to respond to whatever environmental changes happen. So there's a balance between gaining new information, keeping the old information you have, and still making sure you can do your job in cell division and everything. And there seems to be a limit. I don't know what the number is. Don't ask me, Charlie. There seems to be a limit on on certain cell size of how big the genome can be. And the more complicated, you know, I told you that bacillus make spores. There's a whole suite of genes that they have to have resident in them to be able to make spores. So because they are in soil, which gets dry, sometimes you have a drought, sometimes it's hot, sometimes it's cold, they, for the majority, can't lose that. Okay.
Okay, folks, more questions?
Tell me that you're going to think about this when you get home, or you're going to talk about other people among your friends.
I certainly will. Okay, folks. Then we have a special treat for Janet. I have been telling you about how Janet has been, she has a long story with Brazilian astrobiology. She has been supporting us from the very beginning, since the first Brazilian workshop in astrobiology in 2006. And she has always answered our calls. She has come to Brazil 3 times and always giving great talks and trying to engage you, young people. And she really made a significant contribution to the evolution, the arisal and the evolution of Brazilian astrobiology. So on behalf of the Brazilian Astrobiological Society, we have granted Dr. Janet Siefert the title of honorary member of the Brazilian Astrobiological Society. So we have this plaque, and I'm going to read it. Just a second. So the Brazilian Astrobiological Society, SB Astrobio, with its greatest appreciation, hereby awards to Dr. Janet Siefert the title of honorary member of the SBA in recognition to her outstanding and continual support to development of the nascent Brazilian astrobiological community, Sociedade Brasileira de Astrobiologia, from the members of the SBA, Rio de Janeiro, November 22, 2022. Oh. That's for you, so Janet. That's your cue for applause, please. Thank you very much, Janet.
Oh, well, it's been my pleasure. Every single time I've come here, I mean, you are so welcoming always. So thank you. And I cry easy, so don't let this bother you.